Dr. Gallego completed his PhD in bioengineering at the Spanish National Research Council. And he did his postdoctoral work, which is where I met him when he was at Northwestern working with Lee Miller and Sarah Sola. And I think that's where some of the work that we are going to hear about tonight really started to originate, where he started thinking and made some really original contributions to thinking about neurons as populations in low dimensional space when we talk about manifolds. And we will hear a lot about that tonight, so I won't go into any detail about that. Right now, Juan is an associate professor at Imperial College London in bioengineering, but is going to be making a move soon to Champalimaud in Lisbon. Over the years, Juan and I have had some good times together, including a lot of beach time in Jamaica at a neural control and movement meeting, which we'll leave it at that. But tonight, I'm really looking forward to hearing about his talk and an update on what's going on with all the manifold work that he's doing. So tonight, the keynote is sponsored by NeuroXS, so we thank them for their support, but please join me in welcoming Dr. Gallego here tonight.
Thank you, Rob, for this wonderful introduction. And yeah, it's a great pleasure to be here. Thank you for inviting me, by the way, and thanks for coming tonight to see a talk instead of being hanging out outside looking at the mountains. Especially because, I hope I'll repay you by making this, if not a bit inspiring, at least entertaining, because it's like 3 a.m. for me, UK time. Anyway. So I'll be talking about, basically, as Rob said, like manifolds and what they can do for BCI decoding. And I'll be talking specifically about the type of intracortical BCIs that we've been hearing about a lot in this conference and that people who are in this room help pioneer. So basically, you have people with implants and you record the activity from units and multi-units that then you use to control computer cursors or robots or electrical stimulators that reanimate muscles. That said, everything that I'm going to talk about is also kind of informative if you are interested in decoding biomarkers of disease, for example, to control stimulation and do like next generation deep brain stimulation. So let's start from the beginning with the principles of intracortical BCI decoders. And actually the core idea of building an intracortical BCI was there from the 60s and 70s when people realized that when they started to train monkeys to do behavioral tasks and started to look at their behavior. So for example, this is data that I recorded in Chicago, but it's like in the older papers. So you can see the movement of the hand as this monkey was acquiring different targets by moving his arm. And people realized already in the 60s, as I said, that the activity of some neurons in primary motor cortex, that is the main motor area of the brain in primates, looked like the movements of the hand. Like you can see here, it goes up and down. And they also observed that some of them, for example, the magnitude of the response tells you where the monkey is moving to, okay? It's indicated by this arrow. And the nice thing is that another neuron recorded close by would tell you something slightly different about movement and so forth. So then people already proposed in 1970 that if we can record from enough of these neurons, we should be able to reconstruct the movement that the monkey is about to move, okay? And then, thanks to also people in the room developing multi-channel electrode arrays, in the 90s we were able to start doing these experiments where this is a youth array implanted in a monkey. You could record from around 100 neurons at the same time, okay? And now I'm plotting here, basically each neuron is a row and the color indicates how much this neuron is firing, okay? And with this, we can take the behavior that the monkey is moving, generating, so this is the movement of the hand, and you can build like simple linear models and predict the behavior very well. As you can see here, the predictions in pink follow the movement very well. And this was done well before my time and was the basic for great advances in intracortical BCIs, restoring communication in patients by decoding handwriting or speech, and we'll be hearing about that tomorrow. And we've been hearing about this, about restoring movement to paralyzed people by allowing them to control robots or computer cursors, or by reanimating their paralyzed muscles through electrical stimulation, like in this picture. There's also work in controlling bionic limbs, not as cool as in Star Wars, but we're getting there. And there's even people now talking about cognitive augmentation, so kind of downloading Kung Fu. So I think we've made great progress on speech BCIs, and and this is getting very close clinically. What I'll be talking about and what I think our work is relevant for is restoring movement in the sense of trying to restore a rich set of behaviors that we can do, right? Like we can do a lot of amazing things with our bodies. And this is what ultimately I think we should aim for, right? So I've structured the talk in 3 parts that are each related to a challenge that I think we as a community need to tackle. One is achieving stable control over extended periods of time. The other is having BCIs that work out of the box to do many interesting things. So, you do like a little bit of customization or tuning and you can do lots of things. And the last one is, and the most challenging one, which is why it's the last one, is enabling flexible control. So allowing the user to do many, many interesting things, right? That is our ultimate goal. Okay, so I'm going to start with achieving consistent behavior over long periods of time. So the problem here is actually about the signals we record. So intracortical arrays are amazing, and there's ongoing developments, but with current technologies, the problem is that when you record activity, the neurons that you're recording from change over days. Okay, so you can see, for example, here, these are 15 channels from a youth array, and you can see the action potential of each of the neurons that were recorded in experiments a couple of weeks apart. So, for example, you see that there's a few electrodes highlighted in yellow that don't have neurons in one of the 2 days, and then we can use the standard methods to ask whether the neurons that are in the same electrode on different days are the same. And what we usually find in the monkeys that we had in this lab is that after a couple of weeks, around 50% of the neurons that we're recording from were different. Okay? And this number goes down over time. So what this implies is that if you have an intracortical BCI and you have a fixed decoder, if the inputs are changing, the decoder that at the beginning was an amazing tool, gets the job done very easily. After a couple of weeks, it will be like a bit more unwieldy, and after a month, good luck. Okay? So how can we address this problem? So for this, I have to take the Manifoldian detour, as Rob said, and tell you a bit about this way of thinking about the brain that our group and others have been adopting for like a decade plus. So the idea here is that we're interested in how the brain generates behavior. So this is the question, right? So imagine that the classic way to approach this problem is basically to record the activity of neurons, of single neurons, and assume that each of these neurons is the basic computational unit of the brain, okay? But the reality is that neurons are not independent elements in the brain. They have a lot of constraints, such as circuit connectivity and common inputs, that make neurons covary in specific ways. So a neuron cannot do what it wants. It can do what it can do given the constraints that it has imposed by the population. Okay? So the idea of this, you can think about it as you can build a state space, so a space in which each axis is the activity of a neuron in the brain, and the activity doesn't have the freedom to be anywhere. Because of these constraints, the activity will be constrained to a low-dimensional surface that is what we call a neural manifold. I hope this made sense. Yes? Okay. So this manifold captures the potential activity patterns that the brain can generate. Now, there's one additional constraint, that is the behavior the animal is performing. So within this huge manifold, when this monkey is choosing among the 3 fruits in these magical trees that gives you both mangoes, pears, and grapes, so there will be the activity will live in even a smaller portion of this manifold. Okay? So our idea, just to make this clear, because it's the key idea of the talk, is that we think the brain computes or controls behavior based on population-wide activity patterns that lives in these low-dimensional neural manifolds. Okay? This is the theoretical framework. Now, how can we take these ideas and do something with the data that we can record? So now imagine that I've stuck 4 electrodes into this monkey, and I'm recording from these 4 neurons as he is reaching to the different fruits in the magic tree. Then what I'm going to do is build a state space in which each axis is the activity of one of these 4 neurons. Okay? And I'm going to basically project the coordinated activity of these neurons, and it will give me 3 trajectories, each corresponding to grabbing each of the 3 fruits. And I can use dimensionated reaction techniques for those of you who are into these things like BCI, to basically sample this, find a manifold that, if we are right with our way of thinking about the brain, should be an approximation of this true manifold that we believe the brain uses to compute. Okay? Is that clear? And in this framework, then a neuron is nothing more than some projection of what is happening in the manifold. Okay? So now, armed with this theoretical framework, my friend and I guess scientific better half, Matt Perick, who is somewhere here, and I had this idea of how to tackle the problem of stabilizing the decoder over long periods of time. So imagine, again, this high-dimensional space in which each accesses the activity of one neuron motor cortex. Our hypothesis is that when David Bowie was playing E major in that picture, he would always generate the same trajectory in this space, when he was 20 and when he was 60. And we call this true latent dynamics. Okay, this is the hypothesis. And we'll live in some low-dimensional manifold. Now, if we put 3 electrodes in David Bowie's brain when he is like playing E major, the activity of these 3 neurons will describe some trajectory. And if we are right, this trajectory should be a projection of the actual true latent dynamics, right? Does that make sense so far? And now imagine that we wait a few weeks and David Bowie comes back to the lab and we record from 3 different neurons when he's playing E major. Again, if we are right, this trajectory should also be a low-dimensional projection of the true latent dynamics that we can identify with dimensionality reduction. Okay? So now, what is happening here is that both the green and blue trajectories are reflections of the same underlying true latent dynamics. So what is going on, if you can see my hands, for those of you who can, my left hand is the blue trajectories and my right hand is the green trajectories. And from where you stand, these look different. But you can basically find a transformation, a linear transformations or rotations that show you that they are the same thing. Okay? That is the whole trick. We are just finding the transformation that will reveal that these 2 sets of trajectories are similar. And the question is, can we do that? Okay? And we'll learn the latent dynamics. So I'll be talking a lot about this task, so I'll take 2 seconds to explain how it works. So this is one of the classic motor control tasks. So basically, you can think about it as a skill version of table hockey, or whatever this is called, where basically monkeys are holding this manipulandum. They are presented with a target, and after a delay period that we give monkeys so we can look at how they are planning what they're going to do in the future, so they cannot move. And if they move a little bit the joystick, they get a penalty, and this means waiting longer for the juice, so they are very good at waiting. And after this go queue, they move, get to the target, and get the reward. Monkeys are very good at this, and as we have said, we're recording from a bunch of areas of sensory motor cortex during this task. So monkeys are very good at this. You can see here the trajectories of the hand over 2 years. They are like pros of doing this task. We can quantify the similarity, for example, by correlating the profiles across days, and you can see it's perfect correlation. These guys are very good at this task. Now, what we are going to do is test our hypothesis. So we are going to take our neurons, do PCA, so to find a manifold, dimensionality reduction, and we'll get something like this. So this is actual data. These are the trajectories as one of these monkeys was doing this task. Okay? So let's focus on this day. For example, these are the trajectories on day 1. You can see that they are more or less organized by the target. That is indicated with this color-coded clock on the top left. And these are the same, the activity that we recorded from the same monkey a month later. No? So these 2 sets of trajectories look very different. But now the question is, can I do the left hand, right hand, and actually rotate them and show that they are the same? And the answer is that yes, we can. And things become remarkably similar. Yeah? And now I know you may be thinking, oh, these are like, like cartoons. What happens when we quantify things? So this is the correlation across these trajectories in the first 4 dimensions of the manifold. So we get a very impressive 0.6. And I'm saying it's very impressive because if you do a control where you calculate the correlation within within neurons, you only do slightly better. This is the gray dot. Hopefully you can see it. And if you don't do any alignment, your trajectories are very different. And this is true over a month and a half. Okay? So with this simple technique, we can stabilize these latent dynamics. Now the question is, can we actually use it to stabilize a decoder? No, that is where we began. So what we are going to do is like build a simple linear model that predicts how the monkeys move in the hand, like I showed you before. Here you have hand movements for a bunch of trials, decoder trajectories within day, like before. But the cool thing is I can build a decoder today and use it 2 weeks later, and it's as good as the decoder trained on the same day. Okay? If I use this aligned dynamics, and if I don't do the alignment, the decoder is terrible. Okay? And by the way, this was true over a month and a half. So this is within the decoding, and we could do a cross day and use it for a month and a half offline. If you don't align, as you see in the green trace, the decoder basically becomes unusable. We could do this over 2 years for one monkey and 1 year and a half for another monkey. Okay? So we can stabilize a decoder using this simple trick. So we can overcome the problem of neural turnover just by sticking our alignment method in there before the decoder. And actually, a bit after our paper came out, Alan Degenhardt and Bill Bishop, working with Baron Yu, Aaron Batista, and Steve Chase, showed like an online demonstration of the same idea in Nature VME over a few weeks in monkeys. Okay, so this is cool. We can stabilize a motor decoder over long periods of time. But if you are interested in sensory feedback using data from my friend Rai Chaudhry, we could show this for sensory cortex, decoding past movement. And we could also show that we can decode the future movement that the monkey is going to perform during planning over extended periods of time using a stabilized decoder. Okay? And now people have used this for other brain areas, so it seems to be like a universal principle. So stabilizing decoder seems to work. Okay? Now let's move to the next challenge, that is having a decoder that works across different people easily. So again, the problem is very similar, right? We want a fixed decoder, and we want to be able to use the different neurons from different people, so they are able to use the decoder happily, and it doesn't feel like this. By the way, this is an actual sport. There's a world championship of opening beers with a chainsaw. This is, if you want to learn something in this talk, that is a good fact. Okay, so our solution to this problem, or our proposed solution, came from a very simple question, which is, how come that each of us, with our unique brains, can do the same things, right? And our hypothesis is, of course, you may guess by now, that is because we have manifolds that produce similar dynamics, okay? But I'll walk you through that. So basically, the idea is like, we know that if you look at monkey brains, imagine that those are monkey brains. I thought it would be weird to show you actual monkey brains. Even if the neurons that make these brains are unique, the way that these neurons are connected in small circuit motifs and then in microcolumns and layers and regions is similar across monkeys. This is what makes a monkey brain, right? And if you remember, I told you that the manifold reflects constraints such as circuit connectivity. So then you can think that the genome specifies not only the connectivity of the brain, but also the way that the manifolds can look like, okay? So does that make sense, more or less, to everyone? So our hypothesis is basically that the genome encodes the geometry of this neural landscape with manifolds will live. Okay? This is very wishy-washy, but it leads to a testable prediction. That is that if you have two different monkeys with their unique brains doing the same task, if indeed each of their brains instantiates this, if you want, mother manifold, and you record neural activity, find the manifold, then the dynamics should again be the same. So it's the same idea of aligning, but in this case, left and blue are two different monkeys. Okay? So, and I should say that also the similarity of behavior will make these trajectories more similar. Okay? So let's see if this works. Same central data that I've been talking about. Two monkeys, you can see that their behavior is remarkably similar because they were pros. But the neural activity doesn't look very similar because it's two different brains. Even if we do PCA, you see some common features like these rotations that Mark Chochan described in the trajectories, but it's not clear if these are going to be the same or not. So we can do our simple alignment trick again, and what we find is, voila, the trajectories become very similar. Okay? And we can again quantify this. This is the correlation across all 10 dimensions in the manifold. And by the way, this goes down monotonically because how the method works. But you can see that the first few are very high. So now the question again is, in the paper we have a bunch of lower bound controls. If you want, we can talk about those later. But again, the interesting question to me is how similar are these dynamics with respect to how they could be? So we computed the same upper bound that is within monkey, within neurons. And this correlation is not much higher than what we get across two different monkeys. So you can see here, red is very close to gray. And this is actually the case for all the sessions that we compared across three different monkeys. Okay? So now the moment of truth is, does this trick allow us to decode across monkeys? And you can imagine that the answer is yes, because otherwise I wouldn't be talking about it. So this is the position of the hand, again, as before. These are the predictions within monkey, and these are the predictions across monkey that are indistinguishable from the within monkey predictions. And if you don't align across monkeys, the decoder is useless. This is just summary statistics for this session, and this is for all the sessions. So you can generalize a decoder within task across different animals, just, again, using like this simple alignment technique, okay? So this is the trick. Now you stick the alignment, and everyone is happy with their beer openers. But one potential concern about this when we were doing it is like, okay, we're studying center out. What about a more interesting behavior? So we had this data that Matt had recorded where monkeys were presented with one target, but then when they touched that target, another one would pop up, and they had to rush to it, and then another one, and then another one. And they had to complete a sequence of very fast reaches to get a reward. You can think about it as monkey whack-a-mole. It's the same thing as this. So when you look at the behavior, this is how the hand was moved in the workspace. The movements are all over the place for these two different monkeys. So what we did was match movements across monkeys, and the solid and dashed lines are the closest. Oh, interesting. Okay, I shouldn't move. So are the closest movements. Okay. Is this the HTML? Of course it is. Oh, sorry about this. Yeah, no, the battery is fine. It's the HDMI, I think. Oh, it's back.
You have to stand right there.
Yeah, I just okay, and I will try not to move, so we don't. Okay, so yeah, these are the match movements, and again, we could come close to the microphone. We could align the dynamics very well across monkeys, so we could decode across them. But you know, like not everything about the brain is cortex. There's also other stuff below that, underneath, that is interesting, like the basal ganglia. So we are actually now recording in my lab from mice doing a bunch of tasks. But we also had this data from our collaborators and good friends Junchol Park and Josh Dalman at Janilia, where they had trained mice to reach to different locations and pull on a joystick. It's a very cute task. And they were recording from mouse motor basal ganglia, so sensory motor striatum. And we could repeat the analysis. We could align the dynamics, and again, we could decode across mice. And actually, since mice don't love reaching with one hand, there was more behavioral variability. And you can see in this color gradient that the more similar the behavior, which is magenta, the better we could decode across mice. And just to finish talking about alignment across individuals, this also works if there's no movement. So if we look at the moment in which the monkey is thinking about what he's going to do in the future, then we can align the dynamics across monkeys, and we can use the same decoder training one monkey to predict what another monkey is going to do in the future, where he's going to move to. And I should say that we're extending now to more interesting cognitive tasks. This is the work of my PhD student, Joanna Lazar, with Mostafa. I will only give like a 1-minute summary, but this is like an interesting decision-making task where monkeys have to associate cues with future targets and figure out how it works. So we can look at the working memory period and essentially predict the target that another monkey is going to choose based on the activity on a model trained on a different monkey. Now you may notice that there's much more spread than in the motor tasks, right? And this is interesting because in the paper that we're writing, we show that different monkeys solve this task differently, which is why there's like this broad distribution of canonical correlations. So our hypothesis and what turns out is that the more similar the cognitive strategy, more similar the latent dynamics, and the better we can decode across animals. You can see a very strong correlation between the similarity of the strategy and how well we can decode across animals. So this alignment also works for kind of cognitive interesting tasks if you are interested in cognition. Okay. So we've made it to the last part, that is enabling execution of many actions. And this is actually an extremely challenging problem. And there's a lot of, again, going back to the classic papers, there's a lot of interesting classic papers written about this in neuroscience. I want to keep to the center outreaching task for simplicity. And this is a paper from Lee Miller's lab, so my former mentor before my days. And this is the simplest ask for generalization that you can think about. Is the monkey doing the center reaching task with the hand center in front of the right shoulder and in front of the left shoulder? Okay, so it's a very small change, but if you do this with your hands now, if you move like this, or if you start from here, right? Try doing this so we wake up, all of us. So if you move forward from starting on the right shoulder or the left shoulder, you can feel that the muscles you activate are very different. So they did this intentionally because then you could ask, does motor cortex have a fixed relationship with the movement of the hand, like forward, in which case neurons would live in this orange axis, which is no change in the relationship between neurons and movement? Or is it with the muscles, in which case neurons would live in this blue axis, which means that the relationship between the brain and neurons is fixed? So the answer was neither. Each dot is a neuron, and you can see they are all over the place. So this illustrates how hard it is to generalize a decoder, just because the relationship between M1 neurons and behavior is very complicated. We took a stab at this with manifolds, looking at a different data set where monkeys were doing a bunch of one-dimensional tasks. You can see with the wrist, you can see the targets there. But these tasks, again, require different forces and movements. Or as a couple of tasks where monkeys had to reach and grasp stuff. And when we did that, what we found consistently across all comparisons was that even if these tasks were actually quite similar, right, is doing this with your hand versus pushing with your hand, these two, the activity patterns for these two tasks live in different parts of a state space, so different manifolds. Some components of the dynamics were similar, but others not. This is not too surprising. And if we try to find a shared direction that allowed us to decode behavior, we could only decode behavior 50% as well as within task decoder, which is what this red direction means, red bar means. So even if these tasks that are relatively similar, generalizing your decoders is quite hard. And one possibility to go toward generalization could be you initialize like this, and then you use methods in decoder adaptation, and we heard about this from Amy Osborne this morning. And there's other groups working on this, so we could generalize decoders little by little, right? But the problem is if you want people to ultimately do a lot of cool stuff, this becomes very time-consuming. So what may be going on, and what can we learn to make our life easier? So one problem that we've realized and that had been bothering us for a while, and this is a paper by my first PhD student, Katia Fortunato, was that, the linear dimensionality reduction works well, but it feels weird that the brain can be like a linear system, right? Like all these complicated interacting neurons. So what we thought is actually it may be the case that the manifold is very complicated and nonlinear, but if we only look at a tiny part of it, we know that you can linearize locally, right? But if the behavior becomes more interesting, then this linear approximation will break, okay? So this is what we did in the paper. There's a bunch of analysis, but I want to start again with the humble center reaching task. And even in this task, one thing interesting that happens is that if you try to estimate the dimensionality of the manifold using linear methods, you see that as you add more neurons, the dimensionality seems to increase, no? Which means that you are doing a bad job at estimating anything, because once you know the manifold is this, the dimensionality shouldn't change, right? Which is actually what happens if you use nonlinear methods. So this shows that even for this simple task, the manifold is actually slightly nonlinear, okay? And we saw in the paper that you can decode better from a nonlinear manifold. Now, and there's a lot of methods out there that use nonlinear decoding and are better. So now, and by the way, this is like this nonlinearity index, that is the ratio between the two. This was consistent across all tasks. But then we looked at a more interesting data set that was this handwriting data set from Frank Willett's paper with Krishna Shenoy. So this is a human attempting to write all the letters in the English alphabet. And when you look at this data set, it's much more nonlinear than our center-out task. And actually, we can show that if you look at writing all letters, the manifold is much more nonlinear than writing only the subset of letters that look similar, that are closer in the manifold, okay? So manifolds are nonlinear. This is one reason why decoder generalization may be hard. There's a small twist to it that is, again, related to the relationship between connectivity and manifold geometry. That is that different areas have manifolds that are more or less nonlinear. And actually, we found in the mouse task where we had simultaneous recordings that striatum was much more nonlinear than cortex. So, the problem may get harder. Now, I've told you that with this slide, hopefully, that it's clear that one challenge for achieving decoder generalization is that manifolds may have very complicated geometry. Now, the other issue that we're encountering is, as a community, is that actually the mapping between neural activity and behavior is very complicated itself. And there's a bunch of papers on this. One good example is this paper by Juan and Fabio Rizzolio, who is somewhere here, I think, again from Lee's lab, where they had a very interesting idea. That is, okay, what I'm going to do to deal with this complexity is basically record neural activity and look at where I am in the manifold and build a different decoder that will for each part of the manifold. So if you look at this cartoon, there would be like a yellow decoder if you are in that part of the manifold, a violet decoder, a blue decoder, etc. Okay? And what they do during like online is basically you switch decoders depending on where you are. And they use this idea in a very interesting data set where they had monkeys using their hands to do things both in the lab and in a plastic cage that I glued together when I was a postdoc. And it's a super cool data set because some of the actions are similar. Okay? And there's like around 6 actions that they studied. And if you use this trick of finding a nonlinear manifold and using piecewise linear decoders, you can see there in orange this curve that saturates at around 70% variance explained. So they can explain 70% of the EMG variance using this little trick. Okay, so this is quite remarkable, right? And they also show with the DAS red line that if you train like a huge RNN decoder, it does worse than this trick. Okay, and this is again easy to run in real time. But one good thing about this is that we can actually look under the hood and see what is going on. And what they found, and this makes me very happy because it matches Katya's result, is that if you find a flat manifold, everything is kind of like a ball. And if you color code it by the different behaviors, as you see there, they're all like mushed together. But if you find a nonlinear manifold, the structure is much more clear. Okay? But there was like a very thought-provoking observation in this paper. That is, if you look at the exact same task, like it's precision grasping. So this is a monkey doing this with his fingers to grab a little piece of fruit. Look at the colors of the traces. One is orange and the other is pink, which means that these two tasks live in very different parts of the manifold and have a different relationship with behavior, depending on whether the monkey is grabbing the fruit in the lab or in the cage. So okay, there's multiple things that can be going on. I think there's 2 potential possibilities that are the likeliest. One is that there's some cognitive aspect to it that I think is the least likely. There's a second one, that is that we know that there's postural signals in motor cortex that reflect how the animal may be sitting. So maybe the monkey is sitting in a different way in the lab and in his plastic cage. And this is what is pushing the activity somewhere else, even if this is hand motor cortex. Okay? And the third possibility is that the brain may be controlling the spinal cord in a slightly different way depending on this. Okay? So the relationship between the brain and the spinal cord may be changing depending on these adjustments, which makes our life very hard, right, if we want to build a decoder. And actually, there's evidence in a bunch of neuroscience studies that the relationship between the brain and the spinal cord is very complicated, even if you are producing behaviors that are similar. So this is a paper from Andrew Miri, where he had mice either like run on a ladder or grab a joystick and pull on it. So for a mouse, these 2 actions are very similar. And they would just like swap devices. And what they found is like the manifolds are completely orthogonal, which is what this confused color matrix means. So even if it's doing this while you run and doing this when you're reaching, manifolds are orthogonal. Same muscle, similar kinematics. And what happened, which is why I wanted to highlight this paper, is that they use the advantages of the mice. So they use optogenetics to inactivate motor cortex when mice were about to do this. And if you see here, the thing that the arrow is pointing at, if you stimulate when mice are about to reach or pull, muscle activity changes a lot and very rapidly, so 10 milliseconds. Whereas if you stimulate when the mouse is running, the change in activity is much smaller and happens very late. So this suggests very strongly that the way that the brain is driving muscles, like spinal circuits, is completely different between these 2 tasks. Okay? So our life is harder. And the last thing I wanted to highlight is that there's even a cognitive component to this. So this is a paper from Brian De Cleva working with Jen Collinger, where he used this typical trick that my human intracortical BCI friends told me they use a lot,
that is asking their participants to imagine moving their hand, right? I think like people doing this do this all the time, right? And they compare this to when these participants, who were spinal cord injury participants, I think, moved the hand. Okay, so this is intracortical recordings in humans. And you can see that they were asked to create the same target forces, imagine or move. So what they found is that like 60% of the variance of the activity lives in dimensions of the manifold that are common to imagining an action and executing an action. But, surprise, there's a part of the manifold that is for acting alone and a part of the manifold that is for imagining the same action alone. And if you want to decode the action, you need to look at these action-specific dimensions, as you see here. Okay? So to cut a long story short, building a decoder, a biomimetic decoder that maps cortical activity into behavior and works across many behaviors is going to be very complicated. And actually, this is a bit like along the lines of something that my friend Tamar Makin, who is a professor at Oxford, Sam, who is at Yale, and I wrote during COVID over Zoom. We wrote like this little paper thinking about how we could make BCI decoders more generalizable. And if you think about it, there may be like a direction. So far, like for very good reasons, most BCIs rely on motor cortex recordings, right? And this is because we know that motor cortex is the main motor command area of the brain. But we also know that we are missing a lot of areas that are doing interesting computations to drive behavior, like process feedback, or even send outputs to the spinal cord. We also know that the spinal cord does many interesting things. It's not only a bunch of wires. So, we are missing that by only recording from motor cortex. It's even worse because, moving is not the same as acting. Acting requires more cognition. It requires setting your goals. It involves keeping track of what you're doing. And none of these things is very clearly reflected in motor cortex. We know this from neuroscience studies. And if you talk to psychologists like Sam, things get even more complicated because you have to worry about intent. So to summarize, the point that we're trying to make is that motor cortex, we think, unless, neural GPT huge transformed proves us wrong, just building a huge decoder with a lot of data is not going to work because motor cortex has, paradoxically, too little and too much information about behavior. So, too much in the sense that, for example, we may have these postural signals in M1 that prevents us from decoding pinch grasping depending on how you are sitting, in the monkey example. Or in the mouse example, that you are looking at motor cortex and you see these signals and they are actually not driving behavior. They may be just like monitoring what is going on. And too little in the sense that we may be missing important cognitive signals like the difference between imagining and action, okay? And also other higher order processes. So we may need to record from all these areas at the same time. Now there's another approach that I'm not going to talk about because we have like in much detail because we've done very little. That is what some people, including people in this room, are doing. That is taking a different approach in which we take BCI as coaching. You are given a BCI and you are learning to use your BCI. So it's more like learning to play a musical instrument or learning to play sports. And if you talk to a music instructor or a sports coach or a psychologist, they would tell you that the way you learn things is super important to get better. It's not what you learn, but how you learn it, okay? And we have actually a few theory papers that touch on this, and I wanted to show you a slide on it. This is work by Joanna Chang, co-supervised with Claudia Klopath. So Joanna was interested in how what you learn and how you learn it influences your ability to learn new things. So we built a very toy neural network, very vanilla in neuroscience, and we trained this network to do 1, 2, 3, or 4 movements. So very, very simple. First, based on understanding the world, like, there's different angular positions where I can move to, okay? And when we did that, if you look under the hood, you have these nice trajectories in neural space that are each associated to one of these movements, and they, you can make sense of them, right? Like movements next to each other have neural activity patterns next to each other. No? Nice. And if you train a network like this and you ask it to learn a different sensory motor mapping, then the more stuff you know, the better you are at learning new things. Okay? This is all very intuitive, right? This is what everyone would expect. But now let's try to train the network in a different way. So instead of learning an ordered structure, you are learning the network, training the network to say, this queue means do A, this queue means do B, this queue means do C, and this queue means do D, okay? And when you do that, you look at the representations, you see that there's no logic to them, right? You have these points that are there and they are separated, but there's no structure that reflects the structure of the world like before. And if you ask the network to generalize, it just basically sucks, okay? So this is a good example of how, not only how good you are, because the networks were equally good, but how you learn something impacts your ability to learn in the future. Okay, so to summarize very quickly, I've told you a bit about, mostly about the challenges and I think opportunities in motor BCIs. I've told you about how our work and that of other groups, thinking about the brain as manifold, so computation via collective patterns of neural activity can help solve some of these challenges, and also, I should say, answer questions about how the brain works. So I've shown you how you can use this idea to stabilize BCI decoders for basically motor behavior and cognition over long periods of time, or to make these decoders generalize across different individuals, again, during motor and cognitive tasks. We've also talked a bit about the challenges of achieving decoder generalization, which are basically related to these manifolds being very nonlinear and having a very complicated relationship with behavior that, again, we may solve with basically neural GPT, and there's tomorrow a session on foundation models, so we'll see. And the last one is that maybe the approach is just like take learning how to use a BCI, like learning how to ride a bike or play the guitar, and we just need to devise good training curricula with psychologists and people who know about these things. And I had like, Rob, do I have time for 2 bonus slides? So I wanted to highlight a couple of things because I know this has been a bit like a spike chauvinism so far. So I've only been talking about single neurons and multi-units. But there's other signals in the brain, and there's a good case for caring about local field potentials, right? We have lots of recordings and lots of devices that work, and we can record for long periods of time. So this is a paper by Cecilia, who basically was interested in understanding if there's a relationship between the two. So we started with the idea that basically we have neural manifolds reflect biophysical constraints, but these biophysical constraints, or some of these according to people who know about this stuff, like Villan Pesaran, also shape the properties of local field potentials. So what Cecilia found is that there's a strong correlation between some frequency bands in motor cortex and LFPs. You can see this V shape is frequency dependent. And it's fundamental, because if you do this during execution and preparation is the same, and when you try to do it when the monkey is just chilling, the correlations disappear, okay? So this is nice because there's a relationship between these two variables. But there's another thing that is very relevant for BCIs, that we've known for at least 13 years now, that you can decode movement and muscle activity very well from LFPs. So you put these two things together, this means that we can use all our tricks to, for example, generalize the LFP decoders across people. And this is not published, but this is decoding within monkey kinematics. And this is decoding across monkeys. So we can also generalize LFP decoders also over time. And if we don't align, it just doesn't work. So, and by the way, if you want to talk about this, I'm happy to, or about anything. And the second bonus is, what about using neurons from other parts of the body that may be easier to access? So, and there's also been a few talks on, about this in the conferences. So this is work with my also former mentor, Dario Farina, and done by 2 amazing postdocs, Vish and Raji, and Xingchen Yang, and an amazing PhD student, Kira Gibbs. And we are working on a couple of preprints. But what I wanted to show you in one slide is that we are actually doing experiments in which motor complete spinal cord injury participants, and some of them sensory complete, control computer cursors with their motor neurons. Okay? So even if they are motor complete, and in some of them we cannot even see contractions with ultrasound, we can still identify a few motor neurons with surface recordings and doing blind source separation. And even in people with spasticity, so you can see this is the firing of these neurons consistent. If we ask them to do like a attempt doing a contraction, we find some controllable units. And then we can give them cursor control of these controllable units and ask them to generate these interesting profiles. And these are example trials. So they can do this remarkably well. They're actually better than us at doing this. And we don't know exactly why, maybe because the sources are more sparse. But the other cool thing is that then we can be like, oh, after a few days, we were like, oh, we will give you control of a unit associated with another attempted action and see if you can do 2D control, and they can do it. You can see that the first few trials are actually not great, but then they can do it fairly well. Okay? So this is, I don't think this will work as well as an intracortical BCI, but this is non-invasive. And with this, I do want to thank my lab, the funders, and the collaborators. And like as Rob said, I'm moving, so we are hiring. These are the usual architectural pictures of the place you're moving to. It's by the sea. And I also wanted to mention that in addition to joining the Human and Systems Neuroscience department there, I'm joining this new thing that we are trying to create, which is one of the reasons why I was excited to move. That is the Center for Restorative Neurotech that comprises both this warehouse that has like mechatronics and AI engineers, and the idea is to work with companies and clinicians to bring these technologies closer to reality. And also there's a clinic for neurorehabilitation where these patients will be treated and also participate in a study. So if you're interested, just like find me to talk, and I leave you with the final slide, and thank you again. I hope this was fun.
We've got some time for questions, so if you have questions, please step up to the mics and ask away.
Okay.
I was going to ask a question, but then you answered it in that last slide. I was wondering about the connection between single neurons and LFP and these things. So you asked that. So, but a question I did want to ask you is, you've laid out a story which is very clear, very structured, and I'm wondering if you can say something about whether these alignment ideas, if it was in fact the case that like every time you tried it, it worked out just beautifully, perfectly, or were there some hiccups along the way? And can you say something about that?
Yeah, I mean, I think let's come close to the mic. Yeah, so it's interesting because it did work once, and maybe Matt remembers this because I was like, this worked the first time. This never happens. There has to be a bug in the code, but it's like 5 lines of code, so how can there be a bug? But I think what was harder is convince ourselves that it wasn't trivial. And then, of course, convince the reviewers that it wasn't trivial. So we did a lot of controls, for example, so that you can create synthetic neurons and do a lot of stuff, and it just doesn't look as well, or decoders don't generalize as well. For the cognitive story is a bit more complicated, but I think that will require more than the time that we have. And I feel like either this was very clear or everyone is eager to go to the bonfire, or it was very unclear, in which case this is not going to help. Oh, yeah.
We have a question over here.
Super cool talk. Is the mic on?
Yes. Yeah, we
okay.
can hear you.
So I'm trying to relate the two topics of like inter-monkey manifold alignment and intra-monkey misalignment in different contexts. How controlled do you think the environment needs to be for the like between monkey alignment to work? Like, yeah.
Yeah, that is a very good question. And so in the cognitive data set that I didn't, I only showed you like one slide. This was actually, monkeys were chilling in their cage and they were recording basically all their whole behavior, but they could do whatever they want. So we know from the analyzing the behavior that the way they were approaching the task was very different, and some monkeys we could decode from eye movements what they were going to do in the future, from others not. So it feels to be kind of robust. There's also this, maybe I'll try to find a slide because I think this is pretty, if I can find this slide, I think this will also answer your question a bit. So if you can, you can see here. Basically, this is an example of aligning across monkeys versus aligning within monkey in yellow, the different tasks that I was talking about. So I find this like quite striking that you can align better across monkeys than within monkey across these slightly different tasks. So maybe there's, I don't know, this gives me hope. But I think at the end of the day, it's a bit of an empirical question, right? Yeah, because again, if there's these postural signals and so far, maybe it will break the alignment.
Thanks, Juan. Great talk. Can you comment on the role of feed-forward versus feedback control and the role of errors, sensory feedback? I know, big question, but just your thoughts on that.
Yeah, no, this is a great question, and this is something that we're trying to do in the lab. We've designed this this task exactly to do this. It's like a little earthquake machine. So we have mice running, and we push the earth around them, because you need to dissociate sensory feedback and feed-forward control, right? And these tasks, the monkeys were so overtrained that it's hard to know what is feedback and what is feed-forward, right? And even in the mice, unless we do more behavioral work, what we see for decoded generalization is that in the mice, when behavior was less stereotyped than in monkeys, because they don't love unimanual reaching, they were trained for 2 months, not their entire lives, the alignment was worse. So I think this would be a combination of feedback, feed-forward. Now that you mention it, it would be cool to do the analysis in the ballistic phase versus in the corrective phase. So I think we tried and there wasn't a big effect, but we also didn't look very clearly. But it's a very important question. What I would say is that we could piggyback in multi-area recordings to use information about sensory cortex to kind of find motor-only dimensions in motor cortex. Matt has a paper on that. So I think we can use these simple tricks, but it's something that we should talk about, I think.
Rohit.
Hey, great talk. So this might be a little bit of a naive question because I'm still trying to understand what manifold is. But I understand, I think, that across this cognition and different aspects, you're saying that there's a different brain dynamics that you're trying to capture with the manifolds. But in terms of the alignment, you showed that there's like neuron 1, 2, 3 that's doing something in day one, and some different set of neurons doing something like similar things on a different day. What does that mean?
So what it means is that basically you have all these millions of neurons in motor cortex, and they are all working together. So it's like it's like the different players in an orchestra. What matters is the song that they all play together. And if you look at a violin player, a tuba, and I don't know how many more instruments I can think about at this time of night in English, a viola. These 3 guys, if they are playing the same as the other symphony, you can listen to the other 3 guys, and if you know enough music, you will know it's the same piece. So what matters is their collective behavior. So if you sample from enough you can also think about it as in signal processing. If you get enough samples of something, you will be able to recognize it, right? So the idea is like no matter what samples you take of something, if you have enough, you will be able to look at this object. And then you can do all these things of aligning across days. And if the object is the same as we think, you can align across people. Did this more or less help? Okay.
Over here.
Thank you very much for the very nice talk, very interesting and entertaining. So I have maybe a question related to what you just explained is, when you do this alignment across monkeys or within monkeys, how much data do you need to be able to do so? And is it more complicated within monkeys or across monkeys? Do you need maybe more data, more dimension, or is it the same?
It is roughly the same. So the amount of data, you can do this in a few trials per target. And it basically saturates. I don't know if it's in any paper, but we tried at the beginning, and with a handful of trials to each target, you can plateau. Did you ask also about the number of neurons, sorry?
Maybe the number of dimension. Is it more difficult across monkeys than within monkeys, for instance? Or do you need more data to do that, or more dimension in the projections, or something? Or is it basically as easy?
Yeah, no, the short answer is like it's as easy, because I think it's because these tasks are easy. So basically, as I said, like the manifold becomes more complicated as the tasks become more interesting, so you may need more data to align it. But we don't have like tasks that I feel like are complicated enough to actually challenge this. But that would be my intuition, because we also know that to sample the manifold, you need more neurons as the task becomes more interesting. But there's also like an area specificity of it as well. So, but aligning across and within is very similar if you are looking at the same task, yeah.
Okay, thank you.
Oh, yeah. So when you're looking at aligning the manifolds across for a same subject across a period of time, do you notice any trends in the transformation?
Yeah, that's a good question. We haven't done that analysis very carefully to know, also because we didn't have ground truth data of, we didn't know how things were changing too much. And also, even if we were using this statistical method to match neurons, like how much can you trust it, right? It would be cool to have this, I guess, like a side project, if anyone is interested. It could be like now that people are recording in mice or rats with calcium imaging, and you can register the same neurons over days, you could actually ask this question, right? Like, how is the transformation changing? And I'm actually commenting a PhD student working on hippocampus, which is a bit far from me, like looking at a representation of spatial maps. And she's actually trying to understand this, like how, as a mouse gets re-exposed and there's like this change in the representation, how it changes. What we found so far is that it seems close to random. But I don't know. But this is hippocampus, which is weird. Motor cortex would be a bit different, I think. But great question.
We've got 2 more questions over there, and I think after those, we'll finish up.
So I was wondering if you have an intuition of what counts as a different task. So is there like a gradient of manifolds from one task from constrained movements to freely moving movements, or?
Yeah, this is like, I think the $1 million question is how to define a task. Because if you had asked me this question when we wrote this first neuron paper in 2017, I think we said something, I don't know if it made it to the final cut, but that there would be like this huge surface, like I was drawing here, that basically you move along it for all the behaviors. But I feel like, I don't know if this is even a tractable question, because can we ever record from all behaviors? And what we seem to see is that activity jumps from one place to another. So a very dramatic example that made me rethink this, even for a motor domain, is that we have data from monkeys doing center out, but instead of seeing the target, they are presented with a bunch of lines, and they have to figure out where the target is. So if you match the movements from this task, same neuron, same session, same monkey, to the center out, the activity lives in a different part of the state space. So I guess if you take this definition of a same task requires the activity pattern that is the same, then everything seems to be a different task. Maybe because of this dripping of cognitive signals into motor cortex even. But yeah, I don't know if anyone has a better answer, because I think it's a very complicated answer. Just to add to this, I no longer think we can define this based on kinematics or muscle activity, because the spinal cord does a lot of stuff, so I don't have a clue. But if anyone wants to talk about it, I'll be at the bonfire.
One more.
Hello, thank you again for this wonderfully engaging talk on manifolds. My principal question I'm curious about, though, is that this procedure seems to have been replacing other types of pre-processing, such as PCA, CSP, and a few other types of transformations. These transformations are applicable to non-invasive types of brain-computer interfaces as well, and it's not just ECOG. And so my question is, have you tried to apply these manifold procedures to non-invasive BCI modalities?
The answer is no, but I know people are doing it. I'm talking with some. But I mean, like the methods, as you said, can work on anything. We've done it in EMG, if you count it as a BCI. Mm-hmm. So we can align EMG decoders across people, but that is not that surprising, right? Because it's like kind of the same muscles. But I would assume that some of these things will work. For ECOG, at least, like I know Karonesk has some papers, and there's others. So I don't know. I guess like people in this room will know, so it's good that they can come and find you. Okay. Yeah, but thank you.
Thank you.
Let's thank Juan one more time.